Okay, welcome to a biology revision stream. It is getting scarily close now to paper 1. It's the 10th of May today, and it's the 5th of June when we have our first paper. So, I want to go over paper 1 today, and the best way I found of doing this is I have found a load of predicted papers from Primrose Kitten, which has been it's been a decent website I've used in the past to revise. Very expensive website, don't get me wrong, but this is out of 91 marks. It's giving me rough grade boundaries here, things like 65% is an A star. I know that that's not going to be accurate now because the grade boundaries have had massive inflation, but this is 8 questions long. Does it come with a mark scheme, first of all? Or am I just going to have to improv? Okay, it comes with a mark scheme. Cool. So, I think we should give this a little revise just to see what we can remember from paper 1. So, yeah, let's give this a go. Yesterday we went over digestion and absorption, and then we looked very briefly at some statistics, kind of for those questions where it asks you to evaluate something. So it might come up here. Okay, first question is, this is microscopy. Cool. State which type of microscope was used to take the image and explain your answer. Okay. The image, I can't really draw it to be fair. The image is showing a 3D pollen grain. 3D, obviously our two types of electron microscope is scanning electron microscope and transmission electron. Scanning is 3D, transmission is 2D. I always draw kind of a very crude diagram where it shows the electrons hitting the specimen, and in transmission, the electrons go right through the specimen. That's how you kind of tell the difference. Scanning has a lower resolution, transmission has a higher resolution. Transmission electron microscopes, there's a couple issues with them, mainly because the electrons are going directly through, it can cause things like artifacts, visual artifacts. Also, there is a very complicated staining process behind the TEM. Both need to take place in a vacuum. I believe if you wanted to explain this, you would say that it's because obviously air molecules might, bounce into the electrons, causing the electrons to kind of get fucked up. I don't know how you would write that formally, but that would be kind of what it is. In a vacuum, what else? This is like a 1-mark question, but I'm just going into the whole topic here. Obviously the idea that the wavelength of light is too large. Therefore, the light microscopes have a very low resolution. So if you were asked in an exam, explain why this image couldn't be taken with a light microscope, you would say it's because the wavelength of light is too great, light microscopes have a low resolution, electron microscopes have a greater resolution. Always talk comparatively because that is what they're after. So, back to the question. State which type of microscope was used to take this image and explain your answer. So, it's a 3D image. So we would say that this was taken using a scanning electron microscope because the image produced the micrograph, I think you're allowed to say. The micrograph is 3D, therefore it's taken using a scanning electron microscope. I do, man, but look, I have exams very shortly. I'm revising as much as possible. I'll be on tonight at some point, but not right now, then. Oops, that is the other way around. Okay. Calculate the length of a pollen grain. Why is that 3 marks? I can't actually do this because it's online. It's IAM. Image is actual times magnification. Actual is image divided by magnification. Magnification is image divided by actual. We are wanting to calculate the length of a pollen grain. So, you would get the length of the scale bar, which is 4 micrometers. You know what? Let's give this a go. Let's actually try and do this. I have a ruler. Let's try and do this. And this is like the most technical ruler in the world. What the fuck? Okay. According to this, it is something like 7 millimeters. 7 millimeters is equivalent. Let's, just for the sake of doing the math easier, we'll round it to 8. 8 millimeters is equivalent to 4 UM. So we are given the actual, we are given the image. Now we need to convert these. Now, what is it asking for? Is it asking for the actual size? In that case, 4 millimeters, we multiply by 1,000, is 8,000 micrometers. So we are given the scale of, I can't do this in my head. 8,000 divided by 4, 2,000 obviously. So that gives us a 2,000 multiplier. What are we I don't know what we're working out here. Hold on, I'm gonna look at the mark scheme to figure this out. Calculate magnification using scale bar. Length in micrometers or length in millimeters for thousands. Okay, yeah, I can't confirm if I've got this right then. I can't confirm if I've got this right because this is an online paper. And it's going to mess up the scale. That's unfortunate then. I was going to try and actually figure out how to do that. I think you would. You would use the scale bar to calculate the magnification, and then you would multiply the magnification by the overall length. So for 3 marks, that is fair, to be fair. It is saying here, mark point 1 is scanning electron microscope, because the image is 3D. So yeah, we would have gotten 2 marks. Let's draw a little tally out here. So far, I'll give myself 2 marks. We won't give ourselves any marks for that magnification question, because that was a lot of bollocks. I can't answer the question if it's online. You need a ruler, you need to be able to measure it. Okay, please be some nicer questions. High power microscopes must also have a high resolution. Oh, explain what is meant by the term resolution. Okay, wait. Is this the one where you say the ability to distinguish between 2 points on a micrograph? Is that what it's asking? Ability to see 2 objects that are close together as separate objects. Or accept ability to distinguish between 2 objects. Nice, okay. Two marks for what was a very easy question. Pollen is produced... No, not meiosis. Pollen is produced in the male reproductive organs of flowers. A cell from the leaf of a plant was collected. Pollen is the reproductive organ, yeah. That had 36 chromosomes. So its 2n number is 32. Let's quickly go into meiosis. I know the answer, because I did just cheat and look by accident. 36 is 2n. If it's the sexual organ, that means it's a gamete. Therefore, it's going to be n, which is 18. Cool. But we'll quickly go into meiosis, just to explain. The way I figured meiosis to be is it's initially 2n. Then it's, so 2n, and your chromosomes are like this. Then it's 2n, your chromosomes are like this. Then it's n, your chromosomes are like this. Then it's n again, your chromosomes are like this. That's the way I figured out meiosis works. So this is interphase. That is meiosis 1, that is meiosis 2. I have figured out that that is the best way to remember meiosis. 2n, 2n, n, n, line, x, x, line. We're obviously, you know, those are going to be homologous chromosomes. Those aren't going to be, those will just be chromatids, 2 sister chromatids. So yeah, that's how I've remembered meiosis. So yeah, it's 18. I did cheat, but I would have got that anyways. No one in their right mind would have put, like, you know, 72 there. So that's 1 mark. And that's it for question 1. Okay. The diagram shows the mechanism that amino acids can be absorbed through an epithelial cell into the small intestine. Explain 2 adaptations of the cell that allow it to carry out its function. So, 1 is going to say microvilli. We will explain this as small foldings on cell surface membrane, increasing surface area for greater diffusion rate. Your second mark point is going to be there's a lot you could say. You could say contains many channel proteins for greater diffusion rate. The one I like to say is it will likely contain many mitochondria for respiration to provide ATP for active transport. And if you want to be nice, you can say of ions. So many mitochondria for respiration to produce ATP, which provides energy for active transport of ions. Explain why the ileum contains endopeptidases and exopeptidases. Okay. Endo are going to break the end bonds in the polypeptide chain. Exo are going to break the inner bonds. I guess you should say that they hydrolyze. hydrolyze the inner peptide bonds in the polypeptide. And this is 2 marks, so I don't know what else you would say. Microvilli for a large surface area. Yep, so I wouldn't have even needed to talk about diffusion there, but that's 1 mark. Many mitochondria produce ATP to provide energy for active transport. 2 marks. Okay. Endopeptases hydrolyze internal bonds. Exo only remove... Oh, did I get these mixed up by accident? Endo is inner. I don't know whether I said. Exo hydrolyze the end bonds. Endopeptases hydrolyze the internal peptide bonds. Oh, I would have only gotten a mark because I didn't mention the point of that, which is to increase surface area. So, next time in an exam, if you're ever given a digestion question, I did think it. I know I always say that and it sounds like I'm coping. I did think in my head, surface area, but then I kind of thought, why would it need to have a surface area? But no, that makes sense. Yeah, of course. Next question. Describe a process occurring at A. This is showing co-transport. So this is where Na plus ions are moving via facilitated diffusion into the epithelial cell. So sodium ions are moving via facilitated diffusion into the epithelial cell, and they are bringing amino acids through via co-transport using a symport. Scientists discovered this mechanism after treating ileum cells with a respiratory inhibitor. Explain why the absorption of amino acids would stop if the cells were treated with a respiratory inhibitor. Cool, 3 marks. This is nice. Number 1. Number 2, number 3. 3 mark question. Number 1, no respiration means no ATP production. Therefore, no energy can be provided for active transport in the Na plus K plus pump. So, no Na plus concentration gradients can be established. So, no co-transport of amino acids. First one, Na plus ions diffuse into the cell. Really, you would have had to specify down a concentration gradient. I would have put that in an exam. I always say that. Co-transporting amino acids into the cells with them, / each ion co-transports an amino acid. I said amino acids are brought with the sodium ions using a symporter via co-transport. That would be a mark. Come on. So, no mark here, which is surprising. I thought there would have been a mark for saying no respiration means no ATP production. There's a mark for saying ATP provides energy for active transport in the NAK pump, so that's just 1 mark. Concentration concentration of sodium inside cell no longer less. No concentration gradient. No Na plus concentration gradient. No diffusion of Na plus ions into the cell. No amino acid absorption, as this requires the diffusion of Na plus ions. No sodium concentration gradient could be established, so no co-transport of amino acids. I should have gone into that a bit more in depth. But the thing is, with biology mark schemes, you see how, if we assume this to be, everything about that topic, if they want you to talk about this bit, sometimes I will only talk about this bit, the bit before it, and not cover the end. So that is always important, to cover the full process in detail. I would have had to specify the sodium ion gradient is important, so that it can bring sodium ions / amino acids in through the ileum. So far, we've only lost 2 marks out of 10. So that is 10 out of 12, which is 83% so far. Just when I needed to explain a little more in detail, which I definitely would have done in an exam. There's no doubt about that. The fluid mosaic model is used to characterize the plasma membranes. Describe the fluid mosaic model. Shit. I have no idea how you describe a fluid mosaic model. I've never understood what it means. Let's try and improv this. That's what we do after all. Mark point 1, I'm going to say that it is comprised of multiple components, i.e. channel proteins, glycolipids, and obviously phospholipids. I'll put glycoproteins. And these molecules have the ability, it says fluid, that implies movement. I don't know. Let's actually look at the answer for this, because I am going to need it. Mosaic, because there is a phospholipid bilayer with proteins scattered, spread throughout. And fluid, because the proteins are free to move in the membrane. Okay, I would have gotten a mark, I think. I mentioned how it's comprised of multiple components, including glycoproteins. Mosaic, because there is a bilayer. So, let's learn this definition. Mosaic, because there is a bilayer. Mosaic, because there is a bilayer with proteins scattered throughout, randomly arranged. So yeah, with proteins arranged. With proteins arranged throughout. And it is fluid because these proteins have the ability. Proteins are free to move in the membrane. See, I feel like if our class got a question on what is the fluid mosaic, most people would equally have zero idea what to put. Right. Bellatins are water-soluble pigments present in vacuoles of plants. Explain why bennettin cannot cross the plasma membrane. Okay. is it, firstly, I see an ion there. So I'm going to say Benetton is a polar molecule. Second mark point, it is a large molecule, therefore it cannot cross the plasma membrane via simple diffusion. Because it is charged / polar, because it is too large, so it cannot pass through the phospholipid bilayer. Cool. 2 marks. Student carried out an investigation into the effect of ethanol on the permeability of the plasma membrane. They used the following method. State 2 of the variables the student should have controlled that are not mentioned in the method. So 2 control variables. Use a cork borer to cut out the cylinders of beetroot. Rinse the cylinders in distilled water. Use a serial dilution to produce different concentrations. Place a beetroot cylinder in 100 milliliters of each concentration at a controlled temperature for 20 minutes. Okay, wait. So already the easiest things to say, temperature, time, volume, they're all gone. What would we say? Are we allowed to say the mass of the cylinders? Initial mass? And are we allowed to say the species of beetroot? Okay, those are the 2 that I'll go with. Any 2 from variety of beetroot, length, surface area, volume of the beetroot cylinders. I said mass. So let's give ourselves a mark. Let's remember that for next time. Some other ones we could have said is if the skin has been removed, the surface area of the cylinders, the time taken to wash the slices, or the pH. So I would have gotten a mark. 4 marks. Lost 2, 4, 6, 8, 10, 14. So 10 out of 14. No? Is that 14 out of 13? $12 out of 16. No, 14 out of 16. Why am I being so fake? Overall, I've got 77% so far. So that would definitely be an A. Cool. I see a graph question and we can't do this. A, because I don't have any graph paper. B, because I can't be asked. Plot a graph for 4 marks though. That would be a question you would definitely take your time with. The table shows. Would you remove any anomalous results from this? Doesn't look like you would. You would definitely have to calculate a mean though. So you would be showing ethanol concentration on the y-axis. So 20, 40, 60, 80, 100. And on the side would be your mean absorbance value. Wait, has it done the mean for us? It's done the mean for us. That is so nice. Absorbance in AU. Start at 0. You would probably need to go up by 0.1. which means your first one would be there. We'll just plot a very crude diagram here. Obviously use a pencil for this in the exam. 0.3, 0.6. So firstly, it's going to be like this. Then it's going to go up to 0.66. Then 0.67. So that would be what it looks like. Explain the pattern shown by the graph. Let's have a look. So yeah, that is what it's telling us to do. Ethanol on the x-axis, absorbance, linear scale, both axes labelled, all points plotted correctly, line of best fit drawn. I mean, yeah. So, the greater the ethanol concentration, the greater the pigment that is leached out of the cells, which increases the absorbance, because ethanol dissolves proteins in the boiler. So, it was saying explain the pattern. I would have said positive correlation. The greater the thingy-mabob, the greater the absorbance. No, it was not asking you to explain the trend. It was saying explain, tell me why it's happening. And the why would be at greater ethanol percentages, the proteins in the cell surface membrane are going to be denatured, which causes pigment to diffuse out, therefore increasing the absorbance. So we can't give ourselves any marks for that just because, you know, we cheated, but honestly these aren't too bad so far. Hand sanitizer gel works because they contain ethanol. Effective hand sanitizers contain a minimum of 60%, but most contain concentrations higher than 80%. Use the data to explain why. Shit, and I just got rid of it. Well, it was at 20, 40 and 60%, it was still very low. At 80% and onwards, it was greater. So what you would say there is, at concentrations of 60, it is in between, you know, 0.3 and 0.6. However, at concentrations greater, it is more consistent. So it ensures reliability. Below 60%, the ethanol has a very little effect on the membranes. At concentrations below 60%, the ethanol may not dissolve proteins in the boiler. Okay, I wouldn't have gotten a single mark there because it was talking about something completely different. Below 60%, it has a little effect on the membrane. So yeah, below 60%, there is little effect. I would have included data here. I don't know why the mark scheme wouldn't want you to mention data. I would have. Below 60%, yeah, there is absolutely little effect on membranes. Therefore, above 80%, there is some effect on membranes, which would be enough to kill, you know, bacteria, proteins. Right. Final question, and then thing is, I've had like 10, 15 hours sleep today. I feel exhausted. What I might do, to be honest, is I might go and work out for a tad after this, and then I come back later and do another like 40 minutes of revision. Because yeah, paper 1 is obviously about 3 weeks away now. I have next week to revise history paper 1, the week after to revise and then do paper 1, then a week off school, and then it's time. So I might just wrap things up there. 70-ish%, we now know that we should mention more about the fluid mosaic model. We know a little bit more about how to word stuff now. The most important thing is interpreting graphs. If it asks you to explain a pattern, you should always explain the biological impact and not the actual trend. So yeah, I think I will wrap things up there, and I will come back later.